So today's topic is tracer techniques, utilization of radioactive isotopes in biogenetic studies. So here tracer technique means tracer to find out. Techniques are here. These are the techniques which are used to find out the pathway or the steps involved in the pathway are known as tracer techniques. So utilization of radioactive isotopes in biogenetic studies. So these tracer techniques includes the utilization of radioactive isotopes. Generally radioactive isotopes are used in establishing the steps involved in the biogenetic pathways. So what do you mean by isotopes? Isotopes are the atoms of same element with a different number of neutrons. So actually these isotopes means iso means equal, okay, so topes means place. So these isotopes, they occupies the same place, right? Like we have carbon 12, carbon 14, right? So these two belongs to the same element, isotopes of same element with different neutrons. That is, the atomic number will be same, but the atomic weight will be different. Okay, so these isotopes, they have the same atomic number, but they differ in the atomic weight. This is due to the different number of neutrons. So here you can see the isotopes are atoms of same element with a different number of neutrons. So isotopes means equal place. These are the atoms where these are the elements which occupies the same place, but they differ in the atomic weight. That is because of the presence of different number of neutrons. They will have the similar number of electrons, but they differ in protons, they differ in the number of neutrons. Okay, now isotopes of an element will have different masses, okay, and the same number of protons and electrons and the different number of neutrons. Okay, so the atomic number will be same, whereas the atomic weight will be different compared to that of the other element. Okay, so here it contains the same number of protons and electrons, but they differ in the number of neutrons. So isotopes of an element react in exactly the same way because the reactions involves electrons, neutrons, and have no effect on the chemical reactions. So when you incorporate these isotopes into any reaction, like we have carbon, right? So if you replace one of the carbon with the carbon isotope, what happens? So this carbon isotope also involved in the reaction exactly same as that of the original carbon, right? But it will not interfere with the reactions. Okay, so this is because the neutron, they do not have any effect on the chemical reactions. These elements, they differ only in the number of neutrons. Here the neutrons are not involving in the chemical reaction, so it will not affect the reaction, and they involve in the reactions exactly same as that of the original element. So here you can see this is isotope. Isotopes are variations of chemical elements that have same number of protons and differ in the number of neutrons. So here you can see electrons, protons and neutrons. Okay? This is of carbon atom, right? So we identify isotopes based on their masses, whereby sum of protons and neutrons equal to the mass, that is atomic weight. Okay, so the atomic number will be same, whereas the atomic weight will be different because it includes the number of neutrons. So these isotopes, they differ in the number of neutrons, so the masses will be different, atomic weight will be different. So So here we can see we have two examples isotopes of carbon and isotopes of hydrogen. So in case of carbon, so this is our carbon element which contains 6 neutrons and 6 protons, right? And so these are the isotopes C13 and C14. This is C12 is the actual element, and C13 and C14 are the isotopes of our carbon element. So here it contains 6 protons and 6 neutrons. Okay, so the atomic weight, it becomes C12, right? So here we can see we have 6 protons because all the isotopes they will have the equal number of protons. So here the actual element carbon, it contains 6 protons, so C13 also contains 6 protons, right? So now it contains 7 neutrons, so there is a difference in the number of neutrons in C12 and C13, right? So here it contains 7 neutrons, so the atomic weight it is 13, right? So in case of carbon, same 6 protons because the number of protons will be same and they differ in the number of neutrons. So in case of carbon 14, so we have 8 neutrons and 6 protons. In the same way you can see the hydrogen, the mass is 1, it contains 1 proton. And in case of deuterium, so the mass number is 2, it contains 1 proton and 1 neutron. And in case of tritium, the mass number is 3, so the protons are same as that of the original element, so it contains 1 proton and 2 neutrons, so total 3. The atomic weight will become 3. So these radioactive atoms, they are unstable. Okay, so in order to become stable, they will emit the radiations. Okay, so they emit the radiations and try to become stable particles. So this radiations is nothing but, this energy is nothing but radiations. Okay, so this happens in case of radioactive atoms. Okay, so in case of isotopes, we have two types of isotopes, that is radioactive isotopes, unstable, and stable isotopes. Okay, so radioactive isotopes are unstable isotopes. So in order to become stable, they emit the radiations in the form of alpha, beta, gamma and X-rays. Okay, so this is an image of some capsule in the stomach producing radiations, and this is an image of Earth, it is producing radiations. This is how the radiations generally look. Okay, so based on this we will identify the location of the radioisotope. Okay, so the heavy unstable element, it releases or it emits the radiations, gamma radiations, and produces stable compounds. Okay, so definition. So the tracer techniques can be defined as technique which utilizes a labeled compound to find out or to trace the different intermediate and various steps in the biosynthetic pathways in plants at a given rate and time. This is the definition. So the techniques, tracer techniques, from the name itself, trace out means to find out. Okay, so these are the techniques which are used to find out or trace the different intermediates formed and the different steps involved in the biosynthetic pathways, biosynthesis of primary as well as secondary metabolites in the plants, okay, at a given rate and time. So in this technique, different isotopes, mainly the radioactive isotopes, which are incorporated into presumed precursor of plant metabolites and are used as marker in biogenic experiments. So here what we are doing is, in this tracer techniques we are using the isotopes. We have two types of isotopes, radioisotopes and stable isotopes. So here we are using mainly the radioactive isotopes, because these radioactive isotopes, they emits the radiation, so it will be easy for us to locate the or identify the isotopes or identify the labeled compounds. So here in this techniques, what we do is we will first select the presumed precursor. We will select a precursor which is assumed to be involved in all the reactions and also involved in the formation of the product. So here in this technique, we will incorporate the isotope into the selected precursor, that is presumed precursor. So we will incorporate the radio labeled isotopes and then we will insert this labeled compound into the plants. Okay, so when this labeled compound enters into the plant, what happens? So it will start involving in the reactions, same as that of the normal compound. So when this radio labeled compound starts involving in the reactions, so the intermediates formed will also contain this isotopes, radio isotopes. So finally the product will also contain the isotopes if this precursor involved in the formation of the product. So here these radioisotopes acts as marker compounds in the study of these biogenetic experiments. Okay. So here coming to the types of isotopes, we have two types of isotopes, radioisotope and stable isotope. Coming to the radioisotopes, they decay with emission of radiations. Okay, so examples: hydrogen, carbon 14, sodium 24, potassium 42, sulfur 35 and phosphorus 35 and iodine 131. So all these are examples for radioactive isotopes. Okay, so these isotopes, they emits radiations. Okay, so like alpha, beta and gamma radiations. So generally they are used for biological investigations. Okay, so the isotopes used for the biological investigations are carbon and hydrogen, and also used in metabolic studies such as sulfur, phosphorus and alkali and alkaline earth metals. And the isotopes used in the studies of proteins, alkaloids and amino acids are labeled nitrogen atoms, and deuterium is also used in the in the biosynthetic pathways. So we also have the stable isotopes. So these stable isotopes are used for labeling the compounds as possible intermediates in biosynthetic pathways. So here the stable isotopes, they do not emit the radiations. Okay, so in case of radioactive isotopes, they decay with emission of radiation, whereas the stable isotopes, they do not emit the radiations. So they are used in the study of possible intermediates in biosynthetic pathways, and also for the detection of this we have to use the mass spectroscopy in case of nitrogen and oxygen, N15 and O18. So we can use the mass spectroscopy for the identification of the stable isotopes, and in case of hydrogen, deuterium and carbon 13, so in case of these two, they are stable isotopes, so they do not emit any radiation, so they can be detected by using NMR spectroscopy. So in case of radioactive isotopes, we have various detectors like Ziegler-Muller counter and scintillation counter and gas ionization chamber. We can use any of these based upon the radiations. We can select the suitable detector and we can identify this radioactive isotopes based upon the radiations emitted. So this is about the different types of isotopes. We have radioactive isotope and stable isotope. The difference is radioactive isotopes, they decay with emission of radiations, whereas the stable isotopes, they do not emit the radiations. Okay, So both can be used in the study of biosynthetic pathways. So here we have some examples like carbon 14 C14. It is also used in carbon dating of organisms and substances in archaeology, and also to determine the steps involved in the plant photosynthesis. Not only in the photosynthesis, we can also use it in determining steps involved in various biosynthetic pathways. So phosphorus 32, it is used and phosphorus 33, they can be used in research involving biology and genetics. And selenium 75, they are used in protein studies in life sciences. And strontium 85 are used in the metabolism and bone formation studies. And tritium, hydrogen 3, it is used to study life sciences and drug metabolism. So here we can see these are some of the examples of radio labeled compounds. So here in the chloroform, so we have one carbon CHCl3, right? So we are replacing this carbon with the C14 isotope, okay? And we are using this chloroform in several reactions to observe the intermediates or reactions formed, right? And this is one more example for labeled compound. So this is radio labeled methanol. So here the carbon is replaced with the C14 isotope, radio isotope. And this is cymene. In case of this, here at this position 1-methyl, we are replacing this carbon with the radio isotope. And here we have benzaldehyde. So in the ring we are replacing the carbons with the C14. And here also in the ring we are replacing the carbons with the C14 radioisotopes. And here also we are replacing this carbon with radioisotopes, so that we can study the reactions, and the position of this radio labeled isotopes will give you the type of reactions taking place, bond cleavage, bond formation, whatever is taking place in a reaction. Significance. So coming to the significance of tracer techniques, the biosynthetic pathways, the tracer techniques can be used to find out the biosynthetic pathways, like the steps involved in the biosynthetic pathways, or it may be the intermediates, various intermediates taking formed during the biosynthetic pathways, various reactions taking place during this biosynthetic pathways can be found out by using this tracer techniques. So example, by incorporating the radioactive isotope, that is C14, into phenylalanine, the biosynthetic cyanogenetic glycoside pronacin can be detected. This is an example. So like this we can find out various biosynthetic pathways of other compounds, not only cyanogenetic glycoside. We can also find out the biosynthetic pathways of various secondary metabolites. The second one is the location and quantity of the compound containing tracer. So just now we have seen the picture, okay? So like in a stomach we have a radio labeled capsule, right? So like that we can also trace, we can also find the location of this compounds, radio labeled compounds, by so here if we are using any radio labeled compound, methanol some example, right? So we can trace out the location at in which step it is now, okay? So and quantity, how much quantity is consumed and how much quantity is remaining, that can also be traced out by using this tracer techniques. The location and the quantity of the compound containing this radioisotopes can be traced out. So carbon 14 labeled glucose, it is used for the determination of glucose in the biological system. like if you introduce any radio labeled glucose into the body, right? So after the intake of glucose molecule, so the next step is metabolism. Glycolysis takes place, right? So we can also determine the quantity of glucose remaining in the biological system, how much it is consumed, and where it is, and you can also find out the location, right? So we are ingesting from the mouth, and then it is passing through the GIT and then reaching the stomach, intestine. So we can also trace out the location of this radio labeled compound using this tracer techniques. Next, different tracers for different studies. So for studies on nitrogen and amino acid, we can use nitrogen. Okay, we cannot completely depend on the carbon. So we have various radioisotopes. So we have various radioisotopes. So we have different radioisotopes. So based upon the requirement, we can use any radioisotope. Okay, different tracer techniques can be employed for different studies. So in case of amino acids, it includes nitrogen. So we can use the nitrogen radioisotopes for studies of amino acids, like pathways of amino acids can be determined by using different radioisotopes, and it is convenient and suitable technique. The tracer techniques, they are very convenient and they are suitable for all the finding out all types of pathways, or you can also use it for archaeological studies, or also you can also check it in the biological systems. Next, the criteria for tracer techniques. Okay, so the starting concentration of tracer must be sufficient withstand the resistance with dilution in course of metabolism. So here, whenever we are introducing this radio labeled compound, this isotope into the compound, that is your precursor or starting material, so you have to introduce this radioisotope in that concentration. Means the starting concentration of these tracers should be sufficient, and it should be available till the end of this pathway. So if you are using this technique for the study of any biosynthetic pathway, right? So here example, in case of shiknic acid pathway, right? So we have erythrose 4-phosphate and phosphoenolpyruvate, two are there. Okay, so if I am using erythrose 4-phosphate radio labeled compound, so in this, if I included carbon, right, C14, I introduced C14 into the erythrose 4-phosphate. So from erythrose 4-phosphate to till the formation of aromatic amino acids, so the concentration of this radioisotope, whatever carbon 14 isotope which I introduced into this erythrose 4-phosphate, must be available till the completion of this pathway, that is till the formation of phenylalanine, tyrosine and tryptophan. These are the products formed after the shiknic acid pathway, right? So the starting concentration of the tracer, that is radioisotope, must be sufficient, and also it should withstand the dilution. It should be resistant to the dilution, or it should withstand the dilution. It should not get diluted during the metabolism. Okay, this is the first criteria. The concentration, the starting concentration of the radioisotope must be sufficient till the end of the reaction, and it should withstand the resistance and dilution, right? And next, proper labeling. For proper labeling, physical and chemical nature of the compound must be known. So it should be properly labeled. So for this, first you have to study the compound which you want to label with the particular isotope, right? So first study the physical and chemical nature of the compound and then go for the labeling. Okay, so the labeling should be done properly. Next, the labeled compound should involve in the synthesis reactions. So here we are using carbon. Okay, so if I am using carbon C14 radioisotope, right? So it should involve in the reactions same as that of the carbon element. It should involve in the, either it may be the synthesis reaction or it may be breakdown reaction or condensation reaction, whatever it may be. So the radioisotope, it must involve in the reactions same as that of the original element, and the labeled should not damage the system to which it is used. And the most important thing is it should not interfere with the reactions. So it should involve in the synthesis reactions, but it should not damage the system. That means if we are using erythrose 4-phosphate which is labeled with C14, now it is the radio labeled compound, right? So we are inserting this compound into the plant. So this erythrose 4-phosphate should undergo condensation with the phosphoenol pyruvate, right? So the radioisotope which is there in the erythrose 4-phosphate, it should also allow the compound to involve in the condensation reactions, but it should not disturb the reaction. Okay, so whatever intermediates are formed after the condensation should be formed even after introducing the labeled compound. Okay, so the labeled compound, it should not involve in the reactions and it should not damage the system. And the half-life of the radioisotope is also very important. The half-life must be more, then only. So if the half-life is less, like if the half-life of any compound, any isotope is it is around 10 to 15 minutes, right? So the reaction, the time required for the reaction to happen is around 12 hours, right? So in such cases, what happens, after 14 to 15 minutes, this isotope will not be available. It will be reduced to half, right? So it should be available till 12 hours, right? So because the time period for the reaction to happen is 12 hours. So whenever you are introducing the radio labeled compound into a reaction, you should also check for the half-life of the isotope. Okay, so these are the criteria for tracer techniques. These are the points to be noted, checked before starting the tracer technique or before choosing the radioisotope. Okay, the starting concentration of the tracer must be sufficient and it should withstand the resistance with dilution, and the proper labeling should be done. Okay, so before going for the labeling, you should study the physical and chemical properties of the compound to be labeled, and then the radio labeled compound should involve in the synthesis reaction, the general reactions, but it should not interfere or disturb the or destroy the system. It should not cause any damage to the system or reactions. Okay, so this is about significance and criteria, and let us see the advantages. So coming to the advantages, it is highly sensitive. The tracer techniques are highly sensitive and it can be applicable to all living organisms. We can use it in the plants and we can also use it in the biological systems, and the wide range of isotopes are available. So we have seen we can use carbon, hydrogen, sodium, potassium, phosphorus, sulfur, nitrogen. Based upon the requirement, you can use, in case of thio compounds, you can use the sodium, sulfur isotopes. So like this, we have wide range of isotopes. It is not compulsory that we should restrict ourselves to only carbon 14, right? So we have a wide range of isotopes available, so you can use any isotope that depending upon the need, and it is more reliable and easily administered, right? So we have the techniques like direct injection method, and the spraying technique is there, and we also have infiltration method. So the method of administration of this or inserting the radio labeled compound is also easy. Okay, more reliable, easily administered, and isolation procedure is also easy. So we can separate easily. Okay, so these are the advantages. And next advantage is it gives accurate results if proper metabolic time and technique is applied. So the results are also, it gives accurate results. Okay, so these are the advantages. So coming to the limitations or disadvantages, so the kinetic effect, chemical effect, and radiation effect, and radio will be there, and the radiochemical purity. Okay, so based upon the purity of this also, it will interfere with the results, and high concentration distorting the result. Okay, if you use high concentrations, it may distort the results. These are the limitations. So next is steps involved in the tracer technique. So coming to the steps involved, so we have 5 steps. The first one is the selection of radioisotope, and second one is preparation of labeled compounds, and third one is insertion of radio labeled compound in biological system, and the fourth one is separation and determination of labeled compound in various biochemical fractions, and the last one is establishment of precursor intermediate product relation. So the first one is selection of radioisotopes. So in this step we will be selecting the radioisotopes based upon the physical and chemical properties of the compound to be labeled, and based upon the half-life, right, and based upon the compatibility also. And we can also choose we can use carbon, hydrogen and nitrogen. Nitrogen can be employed in case of amino acids, right? So the selection of radioisotopes is very important, right? So for example, if I am using the carbon and hydrogen, right, so in case of sulfur compounds, amino acids, okay, in case of amino acids it consists of nitrogen as well as amino group as well as acid group, right? So if I am using only carbon and hydrogen, right, isotopes, right, in such cases it will be difficult for you to find out how the nitrogen is incorporated, or what are the reactions taking place, right? So you have to think in that angle also and select the radioisotopes. Okay, the selection of radioisotopes, it mainly depends upon the presumed precursor, that is the selected compound, and also the product, right, and half-life of the radioisotope, right? So here we have C14, it has the higher half-life, right? So in such cases we can choose the radioisotope. So while the selection of radioisotopes, we have C11, the half-life is few minutes, right? And C14, it is 6000 years, and hydrogen it is around 12 years, right? So if you select this C14 or hydrogen, in such cases what happens, the half-life is more, right? In such cases they will be available throughout the pathway. Okay, so they are not easily destroyed, or they will not decay easily. It will take 12 years to further decay, right? In such cases it will be available in the plant for 12 years. So the selection of radioisotopes is important. So as we already discussed, same as that of not only the half-life, you should also check for the physical and chemical properties of the compound, and you should also say that it should involve in the reactions, but it should not disturb the reactions. It should not cause any damage to the biosynthetic pathway or the biological system or the reactions taking place, right? second one, the preparation of labeled compound. So once you select the compound, you will be studying the physical and chemical properties of the compound, right? And we will choose a precursor, pre-assumed precursor. So after selecting this compound, we will be incorporating, or we will be introducing this radioisotopes into the compounds. Okay, so after that, when you prepare the radio labeled compound, the next step is insertion of radio labeled compound into the biological system. If I am planning to study the biosynthetic pathway in the plants, now my next step will be the insertion of this already prepared radio labeled compound should be inserted into the plants, right? So if I want to study, if my aim is to study the biological system, metabolism or glycolysis in the humans, right, my next step will be the introduction of this or insertion of radio labeled compound into the biological system, right? So here the third step, here now in pharmacognosy we will be using this for the study of biosynthetic pathways. So now here we are dealing with the plants, right? So first you have to select the precursor. The precursor should be like it should involve in most of the reactions, and it should also contribute to the formation of the product. Then only this radio labeled compound will be there in the product, right? So after selecting the compound, we have to go for labeling using the radioisotopes. Okay, so using carbon or hydrogen, you have to label this compound, and after labeling you have to insert this radio labeled compound into the biological system. Here the biological system is into the plants, right? So after introduction, the fourth step is separation and determination of radio labeled compound. So after introducing into the plant, what happens? This will enter inside the plant and it starts involving in the reactions taking place within the plants, right? So here so let us allow the plant or these radio labeled compounds to involve in the reactions, and then it will form the products. Okay, so it will take certain period, right? So depending upon the secondary metabolite, so we have to allow the plant to allow the radio labeled compound to involve in the certain reactions, steps or stages of reactions, and after that you have to separate the secondary or constituents formed, right? So here we have different methods for the separation, that is nothing but extraction. So by using different extraction procedures, you have to separate these constituents, and you have to extract the constituents and then go for separation, right? So you will get individual compounds. You can also go for the fragmentation, and you can get intermediates also, not only the secondary metabolites or the products. Along with the products, you can also get the intermediates, right? So separate the intermediates and then go for the determination. Okay, so these compounds will also contain the isotopes. Okay, so here we should check for the presence of these isotopes in the products and intermediates formed. Okay, so go for biochemical fractionation, and then take the intermediates as well as the product and go for the determination of labeled compound. Okay, and how will you determine? By using the detectors. Okay, so establishment of precursor intermediate product relation. So during the fourth step, we can identify the compounds, right? So we can identify different intermediates and precursor. So we can find out different compounds formed during this, right? So you have to take all the compounds and you have to identify the precursor. We already used a pre-assumed precursor, we already selected a precursor, so we know the precursor, and we have here we have the product also, because we are aiming at a product, right? So if I want to study the formation of indole alkaloids, then we know that the product will be the indole alkaloid, right? And the precursor will be some phenylalanine or tyrosine or tryptophan, like that. We will be selecting the amino acid, right? So indole alkaloids, they are synthesized from the amino acid. So I select the precursor as amino acid, and my product will be the indole alkaloids, right? So we know the precursor and we know the product, right? So in between whatever components we got during this separation process, we have to arrange it, and we have to establish the relation between the product, intermediates formed, and the precursor. So by like this we have to determine the steps involved in this pathway. Okay, like this the steps involved in the pathways are established. Next, requirement for the tracer technique. So this we already discussed. It's nothing but different steps involved. We have 5 steps, right? So the first step is preparation of labeled compound. So here how will you prepare the labeled compound, right? So we selected a compound, right? First thing is we have to select the compound to be radio labeled, like we need to select the precursor, right? So depending upon the reactions, like in case of acetate mevalonate pathway, okay, so in that we can select the acetyl unit, acetyl CoA as the precursor, right? In shikimic acid pathway, erythrose 4-phosphate or pentyl phosphoenol pyruvate, we can use any of this as the precursor, right? First we have to select the pre-assumed precursor, okay, and then you can use any of these methods and you can label it. So the first method is the radio labeled compound can be produced by growing it in the atmosphere of carbon 14, right? So generally these plants they will utilize the carbon dioxide, and then they involve in the photosynthesis process and produce the glucose molecule, right? So in the same way, these plants are allowed to grow in the atmosphere of carbon dioxide, that is radio labeled carbon dioxide. So in case of carbon dioxide, the carbon element is labeled, that is replaced with the isotope C14 isotope, right? So when you allow the plant to grow in the atmosphere of carbon dioxide, that is radio labeled carbon dioxide, what happens when this plant utilizes this carbon dioxide, right? So the carbon dioxide contains C14 isotope, right? So when this carbon dioxide enters into the plant, so it starts involving in the photosynthesis, that is carbon dioxide and water, and it utilizes the light energy and forms the glucose molecules. So when this carbon it is an isotope, so the formed glucose molecule will also contain C14, right? So like this we can introduce the isotopes into the compounds. Okay, the second method is introduction of tritium, right? So how will you introduce this hydrogen isotopes into the compounds? Like we have tritium, the labeled compounds are commercially available, okay? So here what we can do is we can use the tritium for catalytical exchange, okay? So in presence of hydrogen atom, okay, some reactions will be taking this reduction, okay, hydrogenation. We can go for any of these reactions in presence of tritium. So here example is we can use the unsaturated compounds. Unsaturated compounds contains either double bonds or triple bonds, right? So if you want to convert this unsaturated to saturate, then we have to go for the hydrogenation process. Okay, so generally we will be using hydrogen atom or hydrogen element, then we will go for the hydrogenation, that is unsaturated will be converted into saturated. So here what we are doing is we are using the tritium in place of hydrogen. Okay, so when you use the tritium, what happens, wherever the bonds are removed, okay, the unsaturated is converted to saturated. That means the double, if it is a double bond, if suppose you are taking ethene, CH2 double bond CH2, right? So in such case, if you are going for the hydrogenation of ethene, what happens, the double bond will be removed. Instead of that, CH3 single bond, CH3 single bond CH3 will be formed, that is ethane will be formed. If you go for the hydrogenation of unsaturated compound, that is ethene, CH2 double bond CH2, then if you go for the hydrogenation, you will get CH3 single bond CH3. That means here on both the carbons we are adding one hydrogen molecules, right? So during this hydrogenation, instead of adding hydrogen element, if you add the tritium, that is radioisotope, what happens? So each carbon atom will have one tritium, right? CH2 and one more CH will be there, so that will be your H3 tritium. So on the other carbon also CH2 and one will be the radio labeled compound. Like this also you can introduce this radioisotopes into the compound. Next, by the use of organic synthesis. So by the use of organic synthesis, here you can use methyl magnesium bromide, right? Here we are forming the radio labeled acetic acid. Okay, so here we are using methyl magnesium bromide on reading with the radio labeled carbon dioxide, right? So what happens, one intermediate will be formed, right? So that intermediate, so here we can see this is methyl magnesium bromide. On reading with radio labeled carbon dioxide, this COO is introduced into this methyl group. Okay, so CH3 COO MgBr, CH3 COOH MgBr is formed. Okay, so the COO is because of this carbon dioxide, right? So here we have the C14, so the intermediate formed will also have the C14, right? Now upon hydrolysis, it is giving CH3COOH, acetic acid is formed. Now the intermediate, it contains the radio labeled carbon, so the final product acetic acid, it will also contain the radio labeled carbon. Okay, so by using this organic synthesis also we can by using organic synthesis also we can introduce the radio labeled compound in the selected compounds, radioisotopes into the selected compounds. And we can also use the nuclear reactor. Okay, so by using nuclear reactor or accelerator, right? So here example is N14-7, this on reacting with hydrogen, N14 and hydrogen. Okay, so if you bombard these two in a nuclear reactor, what happens, it forms C14 and one proton. Okay, 14 plus 1 15. Okay, so nitrogen 14 and one hydrogen, total 15 there. Here also we are getting the 15, right? So on bombarding two radioisotopes in the nuclear reactor, so this also leads to the formation of other isotopes. Okay, so like this also we can prepare the labeled compounds. Next, precautions. The precursor should react at necessary site of synthesis of plant, and the plant of the experiment time should synthesize the compound under investigation. The dose given is for short period. So here whenever we are okay, so this is the second step actually. So this is the second step. So introduction of labeled compounds. So now we already prepared the radio labeled compound, we selected the compound and we introduced the isotopes into the compound. Then now next step is we have to insert this radio labeled compound into the plants, right? So now before introducing this radio labeled compound into the plant, the first thing is you have to choose the site. Okay, so where to introduce this radio labeled compound, right? So for example, in case of plant we have the root, stem and leaves, right? So in case of some secondary metabolites, the synthesis takes place within the roots and then it will be carried to the leaves and it will settle down in the leaves. Okay, so it doesn't mean that if the secondary metabolites are available in the leaves, it doesn't mean that they are synthesized within the leaf, right? So there are some cases where the secondary metabolite synthesis takes place within the roots and it will be carried to either stems or leaves and it will get settled there. So first thing is we have to identify the area where the production or biosynthesis of that primary or secondary metabolites is taking place, whether it is taking place within the root or stem or leaf. This we have to identify. Okay, so here the precursor should react at necessary site of synthesis. Okay, so first we have to identify the site and we have to introduce this radio labeled compound into that particular site, right? If suppose I want to study the shikimic acid pathway, right? Assume that it is taking place in the stems, right? So if the shikimic acid pathway is taking place in the stems, right, if I introduce this radio labeled compound into the root, what happens, right? So the reaction is taking place in the stem and the radio labeled compound is there in the root, so it will not be available to involve in the reactions whatever taking place in the stem, right? So the first and foremost thing is to identify the site and you have to introduce or insert this radio labeled compound into that particular site only where the synthesis takes place. The second one is experiment time. Okay, so during 1 to 2 years of age or during this period, autumn period or winter period, the biosynthesis will be there, or during ripening stage. Okay, so it contains high concentration of alkaloids. Okay, like this we'll have the details, right? So the proper time, okay, at what time the biosynthesis of this metabolites is taking place, so that we have to identify, because after the completion of pathway or after the completion of synthesis of the secondary metabolites, if we introduce our radio labeled compound into this plant, what happens? So it will not there is no reaction, right? So it will be there in the plant, right? So the reactions will not happen, right? So first thing is we have to find out the site, and we should also find out the experiment time. Okay, at what time the synthesis is taking place. Okay, the synthesis of compound is taking place in the plants. This we have to identify and we have to introduce the labeled compound into the plants. Okay, so here we have various methods, that is, so we have different methods for introduction of these labeled compounds, that is root feeding, stem feeding. So here you can see the methods: root feeding method, stem feeding method, direct injection method, infiltration method, floating method, and spraying method. So these are the different methods used for introducing or inserting the labeled compound into the plant, right? So we have different methods. We will let us see in detail. So after completing this, we will come to this third step. Okay. So, first one is root feeding method. So root feeding method means, so here we will select the plants in which that particular reaction or the particular pathway takes place in the roots of the plants. Okay, so in such plants, what we do is, here you can see this is hydroponic method, is nothing but root feeding method. So here we will take the hydroponic solution, right? So we will take the hydroponic solution. This contains the radio labeled compounds that to be incorporated or inserted into the plant. Okay, so now the first thing is, you have to select the plants in which the pathway or that particular reaction takes place in the roots. Okay, so this is this method is specific for the plants in which the pathway takes place in the root. Okay, so here what we do is, we have to take the solution, that is hydroponic solution, which contains radio labeled compound, right? Now the next thing is, you have to place the roots of the plant in this hydroponic solution. Okay, so here like this. Okay, so when you place the roots, see here, so this is our hydroponic solution containing radio labeled compounds, right? So when you place these roots intact with this hydroponic solution, what happens? Along with the water, the radio labeled compounds will be absorbed by the roots. Okay, so here the radio labeled compounds enters into the roots. Okay, so this is the first method. Okay, the stem feeding means, here you have to cut a small piece of stem, and then the other part of the stem you have to place it intact with the hydroponic solution. You have to take the solution in another vessel, right? And the plant will remain in the soil, right? So just we have to cut the edge of the stem, and the stem, it is allowed to or be intact with the solution, hydroponic solution containing radio labeled compound. Okay, so we have to take the solution in a vessel separately, and you have to place the stem of the plant into the hydroponic solution, so that the compounds present in it will be absorbed by the stem. That is the second one. Third one, direct injection method. So here you can see, these three are the examples for direct injection method. So here what we have to do is, directly you have to take the hydroponic solution or the solution containing radio labeled compound in a syringe, and like this you have to inject the labeled compound, the solution containing labeled compound, into the stems or bulb, buds or fruits. Okay, so the woody stems are selected for this. Okay, we cannot inject it into the leaves, right? So we can this method can be used in case of buds, bulb, and also fruits and stem. Okay, so directly you have to take the solution containing radio labeled compound in a syringe, and you have to inject it. Here just he is placing the dots on the surface of this leaves, but in case of direct injection you have to inject it into the stems, buds, bulb or fruits of the plant. And fourth one is infiltration. So this is our infiltration method. It is also known as wick method. So here what we have to do is, the plant will remain in the soil itself, right? So here you have to cut small edge of the stem, right? And you have to take a cotton wick. Okay, so this cotton wick and you have to take the solution containing radio labeled compound in a vessel, and now you have to place it near to the plant, and then take the cotton wick and place one end of the cotton wick in the solution and the other end is attached to the plant. So this is called wick method, right? So the isotopes present in the solution will be carried by this cotton wick and it will enter into the plant. So this method is called as cotton wick method. So next is floating method. So here you can see the shallow tray method. So this is nothing but floating method. So here what we have to do is, we have to take the excised buds or leaves or roots or small discs of the leaves, right? So that we have to place it on the solution containing radio labeled compounds, right? So we have to allow the discs to float on the solution containing radio labeled compounds. Okay, that is the one more method. And the last method is spraying technique. So here you can see, instead of spraying they are painting also, right? So directly you can take the sample or solution containing radio label compounds and you can paint it over the surface. Generally you can use this for the leaves, right? And spraying also, you can just take a sprayer and you can spray it. So here he is just placing the liquid using a syringe. He is not injecting, he is just placing. So instead of placing like that, you can use a spraying bottle and you can spray it over the leaves. So the next step is separation and determination of radio labeled compound. Okay, so before going to this separation and determination of radio labeled compound, so let us see the different equipment or detectors available for the detection of radio labeled compounds. So here you can see we have Ziegler-Muller counter, liquid scintillation counter, and gas ionization chamber, and Bernstein-Valentin counter, autoradiography, and radio paper chromatography. All these are used for the identification or detection of radioisotopes. And now we have mass spectroscopy and NMR. So this can be used for the stable isotopes. We have both the radioisotopes and stable isotopes. Okay, the Ziegler-Muller counter, liquid scintillation counter, and gas ionization chamber, autoradiography, all these can be used for the identification or detection of radioisotopes. Okay, and whereas the mass spectroscopy and NMR can be used for the identification of stable isotopes. So now next is separation. That is, first one is, so in case of separation and detection, before going for the detection, immediately after introducing the radio labeled compounds into the plants, you have to allow the plant, right, so that the introduced labeled compounds should involve in the various reactions taking place. Okay, so it should involve in the reactions which we wanted to study, right? So you have to insert in such a way and you have to allow the plant so that the radio labeled compound which is entered into the plant should involve in the reactions or should involve in the pathway and produce the radio labeled secondary metabolite along with the intermediates, right? So now after the formation of secondary metabolites, the next step will be separation. So here we have different methods, like if you want to study the pathway of volatile oil containing drugs, right? And like if you want to study the synthesis of menthol, right? It is a volatile oil, right? So the first thing is you have to separate it, right? So how will you separate the menthol? First you have to separate the menthol, right? And then you have to go for the separation of menthol from the oil, right? So for this you have to use the Clevenger apparatus. You have to go for the hydrodistillation or steam distillation process. Likewise, we have various extraction methods for the separation of secondary metabolites, like we have maceration, percolation, soxylation, right? And soxylation, it is also known as hot continuous percolation. We also have decoction method, microwave extraction is there, and supercritical fluid extraction method is there. We have advanced techniques also, right? So you can use any of these methods and you can get the extract. So after getting the extract, okay, so you'll get a crude extract, right? From the crude extract you have to separate the constituents by using different chromatographic techniques, right? So separate the constituents present in it, and after separating the constituents you have to collect the compounds, and you have to go for detection of radio labeled compound, right? So after separation you will get different number of compounds, right? It can be a precursor, it can be a product, or it can be a intermediate, right? So it can be a byproduct also, right? So the thing is you have to check whether the separated compounds contains the radio labeled compound isotope or not, okay? So this can be done by using the detectors, okay? So here you have to go for the separation by using the suitable extraction method, extract and collect the crude extract, and after collecting the crude extract you have to go for the separation of the constituents present in that particular crude extract, right? So after the separation, the next step is you have to identify, or you have to check whether the precursor, intermediates, products, or whatever compounds you got after the separation contains the radioisotope in it or not, okay? So that can be done by using these detectors, that is Geiger-Müller counter, scintillation chamber, autoradiography, gas ionization chamber, etc. So the first one is Geiger-Müller counter. So it is an instrument used for measuring ionization radiation, okay? So here this Geiger-Müller counter can be used to identify radiation such as alpha particles, beta and gamma, okay? So it can be used for any kind of radiations like alpha radiations, beta and gamma radiations, right? So it perhaps one of the world's best known radiation detection instrument. It has a wide range of detection, right? So it can be used for the detection of the isotopes in the compounds separated, okay? So this is the picture of Geiger-Müller counter, okay? So here you can see we have the monitor, here it indicates, okay? Now here you can see it has a anode and cathode, right? So this is the ionized gas atoms, right? So when the ionizing radiation falls on it, so that will be readed, or that will be taken by this ionized gas atoms, and here it is attached to both the anode and cathode, right? So the readings or the measurements will be given on the display. Next is scintillation counter. So scintillation counter is an instrument. It is used for detecting and measuring the ionizing radiations by using the excitation effect of incident radiation on a scintillator material. Scintillator is an instrument so which emits the light without applying any heat, which emits the light spontaneously, so without applying any heat. Okay, so that instrument is known as scintillator. Okay, scintillator is an instrument which emits the light spontaneously without applying the heat. So here we have a scintillator here, right? So when this ionizing radiations falls on the scintillator, so it starts emitting the light. Okay, so here we have the photomultiplier. It emits the light, okay, that will be taken by the photomultiplier, and then it gives you the readings. So and detects the resultant light pulses. Okay, so this is the liquid scintillation chamber. So here we will place our sample. Whenever the radiations falls on it, it starts emitting the light, and that will be read on this screen. Autoradiography. So in case of autoradiography, so like these images you will get. So wherever the isotopes are there, that location will be traced by using this autoradiography. Okay, so here we will take the sample and place it in the autoradiography. This is the in vitro autoradiography. So when you read the readings, what happens, you will get the imaging. Okay, this is the image scanning equipment. So you will get the image like this. So here you can see the monitor attached to the scanner, right? Here you are getting the images. So if you take the printout, you will get it like this. Okay, so here they zoomed it, right? So on zooming, wherever the isotopes is there you can see. Okay, so here we have the isotope, so the location can be traced out. Okay, autoradiography is the bioanalytical technique used to visualize the distribution of radioactive labeled substance with radioisotope in the biological sample. We can see the distribution where it is distributed. You can find out the location of the radioisotopes in a sample. Okay, it is a method by which a radioactive material can be localized within a particular tissue, cell or cell organelles or biomolecule. So you can find out the location. Okay, so by using this method you can locate the radioactive material in a particular tissue or cell or the cell organelles, or it may be the biomolecule, and it is very sensitive technique, and it is also used for variety of biological experiments, right? And already I told you it is used to locate the radioactive substances. Okay, so it can also be used for quantitative estimation. Okay, so if you see, these are the images of autoradiography. Okay, so this is the plant and these are the autoradiographs. Okay, so here in case of root treatment, here you can see the radiations, and in case of foliar treatment, here we can find the radiations. Okay, so in case of corn, here we have the radioisotope. Okay, and here in the root, during root treatment, so only in the roots you can see the presence of isotopes. Okay, this is how we will locate the isotopes. So this is gas ionization chamber. Okay, so it is an ionization detector which main function is to measure the ion pairs created when ionization mediation through the gas. Okay, so here you can see this is the gas chamber. When the ionizing radiations enters into the chamber, so here it is attached to the battery, cathode and anode. Ionization takes place and the positive and negative ions will be distributed, and it will give you the readings, and that readings can be measured on the monitor. So this is the gas ionization chamber, right? So next is the different methods used in tracer techniques. So next, the methods in tracer technique. So the first method is precursor product sequence. So in this technique, first thing is we have to select the pre-assumed precursor and the pre-assumed precursor of the constituent under investigation. So first you have to select the precursor and then introduce radioisotopes into the precursor, and then we have to insert this into the plant. So this becomes our radio labeled precursor. Now we have to insert this radio labeled precursor into the plant, and after a suitable time the constituents are isolated and purified, and then you have to check the radioactivity. This includes all the steps which we have discussed right now. Okay, so same steps. First thing is select the radioisotope and then select the precursor, introduce the radioisotope into the precursor, and then introduce this radio labeled compound into the plant and leave the plant for suitable time, and after that isolate the constituents, purify it, and then check for the radioactivity, and then you have to establish the precursor intermediate product relationship. Okay, and disadvantages of this method: the radioactivity of isolated compound alone is not usually sufficient evidence that the particular compound fed is direct precursor, because substance may enter the general metabolic pathway, from there may become randomly distributed through a whole range of products. So this is the main disadvantage. So here we are inserting the radio labeled compound into the plant. Okay, so when you are introducing this radio labeled compound into the plant, so there is no sufficient evidence that immediately after introducing into the plant it is going and it is involving in the reactions right away, and it may be the direct precursor or it may be not the direct precursor, or else it may be distributed randomly in the whole product. Okay, so these are the chances. Okay, so either it may be a part of or some other compound may be the precursor, it may not be the direct precursor, or it may involve in the reactions, or it may involve in the reactions from the middle, or it may be randomly distributed in the plant. So there is a chance of all these possibilities, right? So if so, it may not give you the accurate results. So next is applications. It is used for stopping of hardening production in barley seeds. Okay, so the barley seeds or barley seedlings, after 15 to 20 days of germination, they generally produces hardened. Okay, so by using this radio labeled isotopes, you can stop or you can prevent the formation of hardening in the barley seedlings. Okay, that is one example. And you can also go for restricted synthesis of hyoscine. Okay, so in datura generally you can find hyoscine and hyoscyamine, right? So you can restrict only for the synthesis of hyoscine in the datura stramonium. By applying this, you can prevent the formation of hyoscyamine, right? Okay, so this method is applied to the biogenesis of morphine and ergot alkaloids. You can also study different pathways like biogenesis of morphine and ergot-onket. Not only this, we can also study the pathways of different secondary metabolites. Okay, so next is double and multiple labeling. So this double and multiple labeling is nothing but it will be similar to that of the first one. That is precursor product sequence. Okay, so here we can establish the sequence, right? So the method will be same in case of double and multiple labeling, right? So in case of this labeling, either you can use a single compound for labeling purpose, or else you can go for multiple. Double means you can at a time you can label the compound with two isotopes. The same compound can be labeled using two isotopes, like we have seen in the formation of amino acids, right? So amino acids, it includes amino group. Okay, so that means nitrogen. If you want to study the characteristics of nitrogen, how it is formed, then we need to add or we need to use the nitrogen isotope, right? So here we have an example, that is, here you can see lysine. Okay, so lysine, this is the second position. Okay, and 3, 4, 5 and 6, this is the sixth carbon and this is the second carbon. So if you see the structure of lysine, so we have two amino groups in the structure. Okay, so here we have two amino groups in the structure, right? So here if you see, we have two amino groups, one at C2 and one is at C6. Okay, so now this lysine, so it is involving in the formation of anabasin. Okay, so if you see the structure, we have pyridine and piperidine rings. Okay, so here you can see we have two amino groups. So now we need to find out which amino group or which nitrogen is involving in the formation of this heterocyclic ring and which is involving in the formation of this cyclic. So here we are going for double labeling, right? So by using double labeling, we can clearly find out the formation of pyridine ring and piperidine, right? So here we can also find out which nitrogen is involving in the formation of pyridine ring and which nitrogen is involving in the piperidine ring. Okay, so in order to identify that, here we are going for double labeling. Okay, the C2 carbon is labeled with C14 isotope, and here the amino group present on the C6 carbon, we are not labeling this. Okay, so we are labeling this carbon, right? So 6 amino group present at the C6 carbon, C6 carbon is radio labeled with N15 isotope. Okay, here we are using two isotopes. Okay, one is C14. Okay, so C14 we are using at the C2 position and N15 at the C6 position, right? So after the formation of anabasin, if you observe the structure, if you isolate this compound and go for the detection, here you can clearly see this is our C2 position, right? So this is C2 position and this is attached to the C6 position. That means, so this carbon dioxide, this is COO group is removed, and here it is forming a linkage. It is forming a bond. Okay, so the C2 and the amino group present on the C6 is directly involving the formation of piperidine ring. Okay, so in the same way you can analyze in this way also. You can radio label this C2 carbon and also this amino group present on the C2 carbon. Okay, so now we know that this amino group is involved in the formation of pyridine ring, right? Actually here we should have a dot, right? So this is the radio labeled compound. So this is undergoing in the reactions. Actually this is removed and one more nitrogen we are getting, I think. Okay, so here this nitrogen is not involved in the formation of piperidine ring. So the radio labeled nitrogen is not there in the final product. So this shows that only this nitrogen is involving in the formation of the piperidine ring. Next is competitive feeding. So this competitive feeding is used generally to identify when there are two intermediates or when there are several steps taking place during the formation of a product. Like we have A, suppose A is your precursor and C is your product. So during the formation of A to C, it is forming intermediates like B and B dash. Here you can see. Okay, so this is our precursor and this is our product. So during the formation of this A to B, okay, so it is giving A, B as an intermediate, and from B we are getting C. Okay, so from A we are getting B dash and C. So there is a chance that only one of the intermediates are involving in the formation of C. Okay, either B will be involved in the formation of C or B dash will be involved in the formation of C. That we have to find out. So this can be found out by using this competitive feeding. So when there is a chance of two intermediates formed during or two pathways, if it is using two pathways for the production of the product, in such cases we can use this to find out the appropriate intermediate which is involved in the formation of the product. So here you can see this is an example. So here formation of conilin. So here two possibilities are there. Okay, directly from the amino acid we are getting the conilin, and from poly beta keto acid we are getting an intermediate which is involved in the formation of conhydrin. So this is also giving rise to conilin, right? So here this is our intermediate, it is giving both the conilin and the conhydrin. So this such type of reactions can be identified by using this competitive feeding method. The last one is isotope incorporation. Okay, by incorporating the isotopes into the compounds, you can find out it will also give you the information about the position of bond cleavage and bond formation during the reactions. This can be find out by using this isotope incorporation. So if you introduce isotope, radioisotope in the glucose 1 phosphatase, okay, and this reaction is catalyzed by alkaline phosphatase. So this help in the reaction occurs with the cleavage of either CO bond. This can be identified by the isotope incorporation. Okay, so here cleavage of this phosphate group is taking place, right? So that can be identified by using this isotope incorporation method. And next is sequential analysis. So the principle of this method is to investigation of pathways. So here we are investigating the sequence of the compounds formed. So here what we are doing is directly we are growing the plants in the atmosphere of carbon dioxide, that is the carbon dioxide is it contains the radioisotope. So here we are using the carbon dioxide radioisotope which contains C14 isotope, right? So radio labeled carbon dioxide is used here. So in that particular atmosphere we are growing the plants, and then we are analyzing the plant at a given time interval so that we can obtain the sequence in which the compounds are formed. Okay, so simultaneously we are growing the plant in the radio labeled carbon dioxide atmosphere, and then we are taking at a given intervals, we are collecting the plant parts, and then we are analyzing the constituents. Okay, so this will help you to establish the sequence. Okay, carbon dioxide, radio labeled carbon dioxide and sequential analysis has been very successfully used in the elucidation of carbon in the photosynthesis. How the carbon is utilized in the photosynthesis, in which reactions it is involving, you can get it in the sequence. Okay, determination of sequential formation of opium and also tobacco, alkaloids, and exposure as less as 5 minutes is used in detecting biosynthetic sequences. Okay, so you can expose the plants less than 5 minutes in the carbon dioxide, radio labeled carbon dioxide atmosphere, and you can establish the sequence like from piperitone to menthone, menthone to menthol. The sequence can be established in the mentha piperita. So the last part of the topic is application of tracer technique. So we have seen it is used to establish the sequences, study of squalene cyclization by the use of C14 tritium labeled mevalonic acid. Okay, you can study the squalene cyclization. Okay, here the precursor used is mevalonic acid. So we are labeling this mevalonic acid with the C14 isotope and CH3 isotope. And the next is interrelationship among 4-methyl sterols and 4,4-dimethyl sterols by using C14 acetate. Here we are using the acetyl-CoA as the precursor, and we are labeling it with the C14 and developing the interrelation among the 4-methyl sterols and dimethyl sterols. And next, terpenoid biosynthesis by chloroplast isolated in organic solvent by the use of mevalonate. Okay, so we are using the radio labeled mevalonate and it is labeled with the C14, and then we are studying the biosynthesis of terpenoids. Next is study of formation of cinnamic acid in the pathway of coumarin from labeled coumarin. So coumarin is the precursor here, we are labeling it, and we are studying the formation of cinnamic acid in that pathway. And origin of carbon and nitrogen atoms of purine ring. Okay, so here we are using carbon as well as nitrogen labeled precursors. C14 and N15 isotopes are used, and they are inserted into the precursors, and we are studying the origin of the purine ring. Okay, the study of formation of scopolamine by the use of labeled phenylalanine. Okay, so phenylalanine is the starting material for the scopolamine. So here we are radio labeling, that is we are inserting the isotopes into the phenylalanine and we are studying the formation of scopolamine. And next, by the use of calcium. Okay, so you can also use the calcium isotopes, right? So here what we are doing is we are adding this radio labeled calcium oxide or calcium carbonate to the plant, to the soil, right? So that when the plant takes the calcium from the soil, so we can find out the how much uptake is there, uptake of calcium is there by the plants, can be studied. Okay. Next, by adding ammonium phosphate labeled with phosphor 32 of known specific activity, the uptake of phosphorus is followed. Here also we are using radio labeled ammonium phosphate and we are measuring the phosphorus uptake by the plants, and we are also measuring the radioactivity of this labeled compounds. Okay, and we are also estimating whether the lower part of the plant is uptaking the phosphorus first or the upper part. Okay, so and they found that the lower part of the plant is absorbing the phosphorus, and then the upper part, that is branches and leaves, are taking the phosphorus. This can also be find out by using these tracer techniques. Not only the biosynthetic pathways and the steps involved in the pathways, you can also find out like calcium uptake and phosphorus uptake by the plants. So, this completes our tracer techniques. So by this topic the first unit is completed. Okay, so welcome. So we'll see you with the next topic in the next class.